Thank you for the introduction. I kind of threw some pictures in here. I didn't know if we're going to go through the whole introduction, but just some pictures, pretty much what Catherine just said, with some pictures of my animals. This is Superior and Gus, and then my fiance Chelsea down here, and you kind of heard the rest about me. So I will just jump into the article that I'm presenting today. It's called Intertumoral Expression of IL-7 and IL-12 using an oncolytic virus increases systemic sensitivity to immune checkpoint blockade. This is out of Science Translational Medicine. It's an article that I thought was really cool. I had the privilege to work in oncolytics at Mayo, so it's something that I'm kind of familiar with. However, I've never really used the vector that they're using in this study. So it's kind of new to me as well. So before I start with the data and their paper, I wanted to give a little background on the immunology that they talk about. So the cytokines that they utilize are IL-7 and IL-12 in their vaccinia vector. IL-12 is kind of the big kahuna in activating Th1 immune responses, which are key to anti-tumor responses. It's the intracellular pathogens and cancer antigens that IL-12 is really helping with. And then interestingly, they use IL-7, which is not really utilized a whole lot in immune modulatory therapy in cancer that I've heard of before. So we're kind of taught in med school immunology that IL-7 is key to central development of T and B cells as survival signals. It's not really talked about as T cell activation of in the periphery, but it seems like it's pretty key in pushing memory T cells. So that's why they utilize it in this vector. And then later on, once they see some efficacy, they utilize monoclonal antibodies, PD-1, PD-L1 monoclonals, and CTLA-4 monoclonals. These are the commercials that you always see: Opdivo, Keytruda, Yervoy. Kind of dialed back the commercials because it's so effective. It's utilized so often. So most, or not most cancers, but a lot of cancers express PD-L1, which is an inhibitory molecule that binds to PD-1 on T cells, which have intracellular inhibitory motifs that actually shut down T cell responses. So you can see how this is beneficial for cancers to express it. And then in a similar manner, CTLA-4 is a molecule that doesn't have intracellular inhibitory domains, but it just kind of sops up all the B7 necessary for that secondary signal upon binding of antigen MHC T cell receptor complex. And then a little background on the virology. So they use a vaccinia virus, and it is a large double-stranded genome, 190 kilobase pairs, which is really, really big. They have no issue putting large transgenes. They put 2 of them in there and having good expression and not destabilizing the genome. Vaccinia has everything that it needs to copy the genome in the cytoplasm. It doesn't need to enter the nucleus, which is kind of increases the safety profile of it. There's always a theoretical chance if you're putting double-stranded DNA in a nucleus that there could be recombination, but this kind of limits that potential. It's enveloped and it doesn't have a lytic life cycle. So it's not really, so an oncolytic is kind of a misnomer for this. It bleeds off, and a lot of the effects that they're seeing is coming from the immune modulatory cytokines that it's carrying, not actually lysing the cancer cells. And then lastly, it's a member of the POX family. So now back into the article and their data. So the goals of their study were to test their oncolytic vaccinia loaded with IL-7 and IL-12 in a range of different tumor types in vivo. And then once they saw efficacy with that, they wanted to combine it with the checkpoint inhibitors to see if they can break that tolerance. And then they wanted to re-challenge the cured mice to see if that IL-7 really helped with the long-term memory T cell response. So in their intro, they kind of talked about tumor infiltrating lymphocytes, or TILs, within the tumor microenvironment, and they kind of differentiated between hot and cold tumors. Hot being tumors that have a large amount of infiltrating lymphocytes that are getting shut down by those inhibitory molecules that we talked about a couple slides ago, CTLA4, PDL1. And they wanted to induce activation with their cytokine, with IL-7 and IL-12, to kind of turn the tumors hot and then overcome the inhibition with the monoclonals after that to see if they could enhance the response. And then they touched on the fact that it is more immunomodulatory than an oncolytic because it doesn't have a lytic life cycle. And then they mentioned TVEC and Pexivec, which are two FDA-approved oncolytics that encode GM-CSF in a herpes simplex virus. And then they mentioned that their vector is selective for replication in cancer cells to limit the amount of systemic IL-12. IL-12 kind of has a bad rap. It was used a long time ago recombinantly, systemically, and they actually killed some people and made people really, really sick. So it's kind of steered away from as a cytokine in cancer treatment, but it's kind of coming back. But they talk about that they can locally express it, making it a lot safer. So they altered their vaccinia vector, deleted out vaccinia growth factor VGF and 01L genes, which I'm not exactly sure what these are, but I would imagine that they're genes that allow the vector, the virus, to push a cell into cell cycle. Because if a virus gets into a normal cell, it's not going to replicate unless it pushes the cell into cell cycle, and most of our cells are not dividing. So if you delete that out, you can make it cancer specific, because if it gets into a cancer cell that has a defective cell cycle, it's going to replicate freely even without these genes. So that's how they managed to make it cancer specific. And then they modified a B5R membrane protein to reduce antigenicity, which is kind of counterintuitive. You'd think you'd want a lot of viral antigens getting presented in cancer cells to enhance the anti-tumor effect, but you kind of want to mask the virus as best as you can, because you want the antigens being shown on the tumor cells to be tumor antigens, not viral antigens, so you can have that long-lasting memory response. And then they encoded a human IL-7, but mouse IL-12, because IL-7 is cross-reactive between species, but IL-12 is not. And they put it initially in separate vectors, and then they made an all-in-one. They did the separate ones just to parse out the effects that they're seeing from both cytokines. So they did intratumoral injections at 55 millimeters cubed at 3 doses every other day of 2E7 PFU of each vector, adding up to 4E7 PFU of total vector if they're separated, and then just the 4E7 PFU if they were the combined vector. And they did 100 or 200 micrograms of monoclonal antibody at the dosing schedule that is similar in humans. So here are the first results that they got from their empty vector, not encoding any of the cytokines. B16 is a mouse melanoma cell line. You can see they had a pretty good response. This is a really immunogenic cell line. It's melanin is extremely immunogenic, and it's kind of cool. If you have a really good response, you can actually see the skin and the hair, the fur of the mice bleach if you have a really good immune response to melanin. And right where that tumor was, you can see a white spot, which is really, really interesting and cool. CT26 is like the gold standard of mouse cancer models. It's really difficult to treat. Not a lot of tumor antigens in this model, but they're still seeing pretty good effects here. And then LLC is a mouse lung carcinoma. So here's the tumor progression graph of the separate cytokine loaded vectors. You can see most of the effect, anti-tumor effect, is coming from the IL-12 when you separate it out. IL-7 is not doing a whole lot here, which is kind of to be expected. IL-12 is a really potent one, but then when you combine them, you can see 4 out of 7 completely cured, which is really good. So then they looked at the immune profile within these tumors, and they showed an increased number of CD8s, natural killer T cells, and then unfortunately CD4 FOXP3 Tregs also increased. So a lot of these T cells are getting in there and then getting shut down and pushed into the Treg lineage once they encounter all of the inhibitory molecules like CTLA4 and PDL1. They didn't show any change in the percent of Tregs of total CD4s, and kind of to be expected, they saw an upregulation of PDL1 because they're having a lot of cell-mediated death of all of the tumor cells that are not expressing PDL1. So you're selecting for those ones that are. Then they did a nanostring pan-cancer immune profiling panel. So I threw this in here. I didn't exactly know what innate meant or adaptive, so it's kind of arbitrary. But it looks cool, I suppose. Then they increased interferon gamma, which makes sense by just the cytokine pathway of IL-12 being expressed at high levels. You're going to push macrophages or T cells to make interferon gamma to activate macrophages. And then the IL-12 groups upregulated MHC class 2 on M1 macrophages. So then they did a model of metastasis by doing another tumor on the opposite flank and only treating one. Interestingly, they found that no viral genome was in the distant tumor, indicating that if they saw any effect, it was purely immunogenic, not the virus going through the blood, getting to the other side. It was the immune cells going through the lymphatics and then raising a response to tumor-specific antigens on the other side. They did see some viral genome in the LLC model, which they did not have a rationale for. So once they saw that efficacy, they moved into the combination studies. You can see that there was no response in CT26 tumor model, either distant or injected tumor from the monoclonals alone. This one right here is you're seeing good responses from the combination or the combined vector alone, no monoclonals, but you're not seeing a great distant tumor response, only 1 out of 10 cured here. However, when you add the monoclonals, you have that same good response in the injected tumor, but then you're getting 6 out of 10 and 4 out of 10 completely cured in the distant tumor, which is phenomenal. And then they're seeing reduced percent of Tregs of total CD4s in this instance with the combined monoclonals and oncolytic. So then they moved into rechallenge studies. They took the splenocytes from those cured mice and they looked at interferon gamma release as expected, very high expression of interferon gamma levels. And then they took those same mice and challenged them. I think it was 6 months after the initial treatment with the same tumor type. And then these are age matched, treatment naive mice. You can see the tumor engrafts in these grows really well. And it was completely rejected in all 10 of the mice that were previously cured with the oncolytic and the monoclonals. So they wanted to test the robustness of the memory response. So they took those previously cured ones and did a intracranial challenge. And you can see that the tumor was again rejected even in the cranial cavity. So um, uh, it's pretty hard for immune cells to cross the blood-brain barrier. And if you're seeing a response in both of those spots, it's um, pretty good. And these are tumor cells that express luciferase. And then you inject these mice with luciferin, and then wherever that tumor is located, it luminescence, and then you image it with an IVUS machine. So that's what those are. So then they made a humanized model of this, probably to eventually move into humans. So they had to swap out the mouse IL-12 with human and make that vector, and then they tested it in some human cell lines. Obviously, you can't put human cell lines in mice, so you have to use immunocompetent nude mice. However, when you do this, these cytokines are irrelevant because there's no T or B cells in these mice. So you're only measuring the oncolytic activity in this model. And it's kind of exaggerated as well because they don't have an immune system. So the virus is able to replicate unchecked and continue to replicate and kill all of those cells. So it's not the best model, but it's something that for oncolytics you always have to do before you go into humans. So then they took A549 supernatant that has all those cytokines produced by the vector, and they used it to stimulate human peripheral blood mononuclear cells, and they showed proliferation and differentiation of those monocytes into macrophages, and then this is just showing the amount of interferon being produced. So in their discussion, they kind of talked about the pros and cons of their platform. They mentioned how the vaccinia replication cycle is really short, which minimally exposes viral antigens and kind of puts a spotlight on the cancer antigens, rather. So they think that that's a good reason for their platform. They talked about upregulation of PD-L1 and how it's typically bad. But now with the monoclonals, you can target it and you can break tolerance fairly easily if you can turn those tumors hot. And then they want to do a panel of biomarkers to identify susceptible tumors and kind of identify the antigens that they found and made good memory responses from. That would help not only them, but other researchers looking at these tumor types and identifying antigens that they're mounting good responses to possibly make cancer vaccines or other treatments. So that's all I have. If anyone has any questions, I think the paper, if anyone's interested, will be posted on the website maybe. So thank you.